Hi class. Now we are going into a new chapter, which is called as Biotechnology and Medicine. This is a chapter which is a collection of small, short topics, and each topic is very important, and it is more important from an exam point of view. So pay attention. It's very interesting and important. So as I mentioned to you, the chapter is called as Biotechnology and Medicine. And I have described to you the meaning of biotechnology, where fundamental concepts from biology, if they can be developed into a technology, and this technology could be useful globally in solving human health and diseases. For example, as I always mention, insulin using bacterial products, recombinant DNA technology, is one of the most salient products of this technology, and it helps in the treatment of disease such as diabetes. Also, generation of genetically modified plants, animals to increase their life, to increase productivity, increase commerce. So, but this chapter is a collection of short articles, and so the first topic rather is disease diagnosis. So what is disease diagnosis? So we all get unwell, and we generally call ourselves sick. Now this sickness could be, let's say, if you are getting sick in this climate, in this situation of this pandemic, and let's say if you have a sore throat, let's say you have a tightness of chest and fever, then with these three situations, we will doubt ourselves to be having COVID-19, right? But before this COVID-19 pandemic, if this would have happened, you would consider this to be a seasonal flu, right? So now, if in today's climate you would like to get ourselves checked up by let's say by doing RT-PCR for looking at active infection, right? Or we could do antibody test for looking at both active as well as prior infection. Right? So now, at this point of time, it's a dilemma, right? That's whether somebody has flu or you have exposure to coronavirus, and both have very much overlapping symptoms. So one would like to walk into a clinic, a diagnostic lab, and give blood or saliva or nasal swab, whatever it takes. We want to know what exactly is going on. Now, to find the cause of sickness is extremely important. Finding the cause of sickness is extremely important because once we know that the cause, the treatment will be effective. Cause is not right. Cause has not been determined. The treatment will not be right. So we cannot treat flu with COVID-19 medication, whatever we have available, and vice versa. So diagnosis is that process which helps to establish the cause of a disease, because unless a cause is established, the treatment will not be right. All right? I hope you agree with me. So that's very important. diagnosis. Now, this diagnosis could be very straightforward, right? Or it has to be very, very methodical. It has to be very much logical, because a wrong treatment could lead to major complexities, right? And diagnosis is not recent. It has been done since in Egyptian civilization, traditional Chinese civilization, and so much so that in those days there were physicians, let's say, doctors in those days. They could treat patients or sick person by looking at the counting the pulse also. So it is not new, it has been, and it has evolved constantly in the light of technology. So diagnosis is the procedure to establish the cause of a disease. Now, if somebody has a fracture, then definitely he or she has to go to check the structure of the body, structure of the bone. So that's anatomical description or anatomical diagnosis. If somebody is having a change in, let's say, sodium ion or potassium ion, then they have to go for a biochemistry test, look at the sugar level in the blood to look for diabetes. So those becomes physiological diagnosis, right? Pathological diagnosis, let's say when we give urine or when we give blood, or sometimes a biopsy is done to find out the results, whether somebody has a tumor or non-tumor, or there is what is the anti-cancer medication is working. So we have to go for pathology. The most emerging disease of modern times is psychology. And stress, the metropolitan stress, the pressure, it is causing a lot of problems. And so this is an emerging field that will continue to grow as our life is changing. So we need to know what is going on in terms of our brain, our hormones, our immunity. They're all complex together. So psychology would like to get into mental health, behavior, social presentation, and how we interact. So there are various angles, there are various arrows that points towards various diseases, and each disease has its own treatment. It cannot overlap, right? A good knowledge of this will make somebody a good paramedical person, a good nurse, a good doctor, a good scientist, physician scientist, right? So diagnosis is very important. That's the bottom line. Right? Now, diagnosis sometimes could be very easy. It could just depend upon the case history. Let's say if an allergy season, and let's say there was not a pandemic era, so if somebody goes with red eyes in the allergy season, then the doctor or physician will prescribe anti-allergy and the anti-allergics to that person. It could be, let's say, it could be Allegra or Claritin or like this. So, but there are some situations where a doctor would like to monitor you. So they'll say, okay, took this medicine for 2 days. If it doesn't work, then call me. Then we will change the dose or increase the medicine or change the medicine. So there will be follow-ups, right? And then doctor at the end of a week or 2 weeks, they'll try to assess whether it is working or not. So it's a process from very simple, giving the medicine and just by listening the history, or it takes time by multiple follow-ups. Now, there can be various types of diagnosis. The most simple, I won't say anything as simple, but the most straightforward could be a clinical diagnosis, right? A clinical diagnosis is when the clinical diagnosis is when the patient walks into the clinic and the symptoms align with what is happening in the society. For example, flu season or cold season. So a physician already has a pre-knowledge that that something is going on in the society, in the population. So he can readily give a anti-allergic medicine, as I mentioned before. In today's date, if somebody goes with sore throat, fever, chest tightening, so doctor will definitely want to rule out whether it's COVID-19 or something else. So it is simple, but in today's date, it is complicated to diagnose something like fever or sore throat or itchy eyes or running nose. So as we learn, we will learn more about COVID-19, and then we can make the diagnosis and the treatment more easy. So this is clinical diagnosis, where a physician, essentially by looking at the clinical history, can come up to diagnosis and start treatment right away. The second type of diagnosis is lab diagnosis. So let's say lab diagnosis. So let's say a patient comes with a clinical picture which is not clear what the person is going through. So a physician needs to send samples to lab. So a physician or a doctor needs the help of a lab, right? And in the lab, the samples that could be sent is blood, urine, stool, saliva, right? So a physician is confused and is not ready to diagnose clinically based on symptoms. He or she needs to send these samples to Quest Diagnostic or LabCorp for diagnosis. Until that diagnostic reports come back, he won't declare the disease. He won't share the disease. So he is still waiting. So when a diagnosis is dependent upon lab report, that is called as lab diagnosis. The third kind of diagnosis is called as radiological diagnosis. So a radiological diagnosis is that kind of diagnosis where the physician wants to look at the image of the body, right? He wants to see what kind of fracture it is. He wants to see how deep the tissue is damaged, how the fracture is simple or complex, right? He wants to see whether the tumor has regressed after treatment. So in all these situations, he wants to take the picture of the body parts. So that is called as radiological diagnosis. Right? Sometimes cancer patients are given radiation rays for treatment. So those are also part of radiological treatment. This is radiological diagnosis. Okay? So we discussed clinical, lab, and radiological. The fourth one is principal diagnosis. When a physician has done all the tests, all the necessary tests to come to a conclusion that this is the main diagnosis, and from here, the treatment should start. That diagnosis, that word, or that sentence becomes the main diagnosis, and that becomes the starting point for the treatment. A treatment could be just simple medication, it could be surgery, it could be many things. And if there is a situation where a patient needs to be admitted for more follow-up diagnosis or to start treatment, that diagnosis is called as admitting diagnosis. So this is one of the first small topic called as diagnosis. The second topic is called as gene therapy. So gene therapy, as the name itself suggests that gene comes from the word DNA. So when DNA is used as a treatment modality, right? This treatment is called as gene therapy. Now, why? So this could happen in some diseases where the nucleotides in the DNA is altered. When nucleotide is altered, it could give rise to a abnormal protein. And for example, in sickle cell anemia, where a single nucleotide change disrupts the structure of hemoglobin that we discussed in proteomics, and hemoglobin cannot change its function. A sickle type hemoglobin cannot deliver oxygen and carbon dioxide efficiently. So a person with this mutation will have problems, and they end up going for repeated transfusion, and repeated transfusion increases the chance of infection. So it has a heavy toll on the patient. So DNA is abnormal, so DNA has to be replaced. So to replace a disease DNA with replace a disease DNA with a healthy DNA, right? And this healthy DNA is then is put back into the patients, right? So the abnormal DNA has been replaced by healthy DNA, and this healthy DNA has then again put back into the body of a patient, right? So if it's a sickle cell anemia hemoglobin is dysfunctional. So the new healthy DNA has to be put into bone marrow and has to assure that this bone marrow is making a healthy hemoglobin. So when DNA as a molecule is used as a therapeutics, that is called as gene therapy. Now the problem here is, the challenge here is that is a delivery. So DNA is a double helical molecule. Now this DNA molecule, let's say it has to enter into the bone marrow. So this DNA molecule, now it has to enter into the bone marrow such that the DNA that is inside, the DNA that is abnormal is replaced by the healthy DNA. So this is very, very challenging. So how to take care of this? So what scientists have done, you need a vehicle to transport healthy DNA into the bone marrow of a patient who has a mutation by which his hemoglobin is sickle shape and has problem. So we need a vehicle. So the most popular vehicle for gene therapy is a viral system, and you will be surprised here that for recombinant DNA technology, we use bacterial plasmid, bacterial restriction enzymes, and for gene therapy, we use adenovirus. So despite these viruses, which are naughty in terms of creating disease, they are being used by scientists as a tool to deliver or to treat disease. So now, how is it possible? So you can ask me how it is being done. So let's say in adenoviruses, there could be, there are some, so let's say this is the DNA of adenovirus, right? Now this adenovirus could be made up of a gene, DNA, which are bad. So there are patches of bad DNA in this adenovirus DNA. So what scientists do, they will, using a restriction enzyme, they will cut the bad DNA, and then after cutting the bad DNA, they'll remove the bad DNA. Bad DNA means that causes disease. So scientists using restriction enzymes will remove the bad DNA, and they will have only good DNA left. So this adenovirus, which has been removed, which has now independent of the bad genes, can be used as a vehicle to transport the good DNA, the healthy DNA, into the bone marrow to treat sickle cell anemia. So this, in short, is gene therapy. So let's go back to the slide. So here, this is an adenovirus. The adenovirus gene is being taken, and the bad genes are removed, and the good genes is put into this vector, and then this whole plasmid is again put inside the patient. So adenovirus is one of the most popular strategy for gene therapy. So this was the second topic of small topic, and gene therapy is not very old, and there are success in gene therapy, particularly in blood cancers, in a disease called as SCID, where patients have problem to fight infections. They lack one enzyme due to mutation. So, and it's also in leukemia, multiple myeloma, gene therapy is relatively more successful, but again, it depends upon the success of gene delivery, and then transport of healthy cells into patient's body, and patient's body should be able to accept that. So as these technologies are very important, but they have risks. So right, so adenovec, there are various strategies that I won't go into detail now. Now, vaccines, everyone, we are growing in a climate day to day when we are waiting for vaccines, right? Especially in time of this pandemic, and everybody is hoping that by this year end or early 2021, we have a vaccine in our hand, so we can go back to a normal life, right? And schools open, jobs open, everything. So what are vaccines actually? Vaccines are those molecules that that boosts our immune system. Why to boost? To fight infection. Right? Now infection. Nowadays vaccines are also against cancer, not necessarily to fight infection. Now, how does a vaccine boosts immune system? So, for example, let's consider one of the most successful vaccines called as polio vaccine. Yeah. Polio vaccine or flu vaccine. Right? So, in here, the polio vaccine is injected very early in the life. Maybe first few weeks or first few days, very early after birth. So this vaccine, first it used to be injected, but now it is given orally. So that is also a development, because if you inject a small baby, it's painful, but if it is given orally, it is more. So vaccines are advancing in that direction also. So when a vaccine is delivered into a baby, these baby they have a special they lead to the development of special cell called as memory B cells, and these memory B cells they work as a depot effect, such that when this small baby develops into a kid, teenager, adult, and come across polio virus vaccines, he or she has these memory B cells. These memory B cells will produce antibodies to fight the new infections, right? So vaccines are those agents that are there to boost immune system, and this immune system serve as a memory, and when the person encounters this bug pathogen again, then these memory cells are ready to fight. They don't have to educate themselves again, they are ready to fight. Now, there are some vaccines. So there are various stages of development. First, the vaccines are very crude, and then then it is modified, with the help of technology. So with the reason for vaccine development is to increase the production, reduce the cost, and make it globally available, right? So let's say there are some places in very corner of the globe where people may not have refrigerators. So how to freeze, how to put it in fridge, Even if they have a fridge, they don't have 24 hours power. So how to make vaccines more robust so that they can tolerate extreme changes, right? So one thing very interesting I want to mention that there could be two kinds of vaccines. One is prophylactic. It is given before, so let's say it's given early in the life, such that we have memory B cells that can counter infection when the bug again attacks. The second kind of vaccine are therapeutic vaccines, and this is in cervical cancer. Uh, Human papilloma virus causes cervical cancer, and this so cervical cancer is caused by cervical cancer is caused by HPV, human papilloma virus, and this HPV vaccine can be given to a cancer patient, as well as it can be given as prophylactic, such that people don't develop an infection. And also, so HPV vaccine is two, prophylactic as well as therapeutic, whereas polio vaccine is mainly prophylactic. Okay. So what was the first topic? Diagnosis. Second topic was gene therapy. Third topic is vaccine. So let's see what's the fourth topic. Regenerative medicine. Regenerative medicine is mainly like stem cells. Some hospitals now, they have a department like tissue engineering. They also have biomedical engineering. They have stem cell biotechnology. So they're all part of medicine to replace the diseased part of the body with the healthy part, such that a person can function normally. So this is a very simple, just on the lines of stem cells to replace the diseased organ or the diseased tissue. Next very important small topic is ribozyme application. So, excuse me. So let's say, so I mentioned to you this pathway that DNA can replicate, or it can transcribe or translate into RNA, and then RNA can translate into protein, right? RNA translates into proteins. Now, think about this situation, that if the DNA is bad, right, the RNA will be bad. If RNA is bad, then the proteins will be bad. So because of this, the whole cellular program will be affected. And then it's difficult to counter or treat the person. So a DNA can be treated. If the DNA has mutated, it can be treated by replacing with healthy DNA, and this process is called as gene therapy, right? A protein could be replaced. For example, let's say Fabrizyme that I talked, it can be supplemented. So that could be called as a protein therapy, right? So if protein is bad, then we can put extra protein or a normal protein. But the question I'm getting to is how to treat the diseased RNA, how to take care of this. So there is a recent technology. This technology is called as ribozyme technology, and ribo, one word, and the second word is zyme, and zyme comes from enzyme. So when RNA acts as an enzyme, this product is called as ribozyme. Now this ribozyme, if RNA is bad, this bad RNA can be destroyed with the help of the ribozyme. So DNA could be substituted by gene therapy. Proteins could be could be taken care by protein therapy by providing more protein, normal protein. Ribozyme is the process where the RNA, so let's say this is a disease mRNA that would make diseased protein that would exacerbate the disease. So these ribozymes will break the RNA, cut into pieces, so that the bad protein is not made. And this ribozyme was discovered by Thomas Cech and Sidney Altman, who won the Nobel Prize. Right? Then the second topic is genetic testing, which is very important. Nowadays, you see that there are many testings that are available there, which says that submit your chromosome DNA and we will look at any genetic diseases. So it is coming to our home. We don't have to go. So they will provide a kit and we put our saliva or we put our urine and they will look at the whole picture inside our body and will depict whether there is a chances of developing cancer. So genetic testing for reproductive reasons and all those is very important. Now the last small topic that I would like to discuss today is called as Human Genome Project. Human Genome Project. And this is the most important project of modern days. And as the name suggests, human genome. So we talked about proteomics, proteome. Here we're talking of genome. So when we look at, so this is an approach to look at the sequence of all the DNA on 46 chromosomes. So I mentioned to you, right, that there are 40 in our cells within the nucleus, there are 46 chromosomes, or there are 23 pairs of chromosomes. And these chromosomes are made up of DNA plus proteins. And our here, the human genome focuses on DNA, reading of DNA. So what are the alphabets in DNA? There are only two, A, T, G, C. So now this chromosomes, if it is the DNA is elongated, it could be 6 meters long DNA. So how to read it, especially these alphabets? So you need a very sophisticated approach. So that project was called as Human Genome Project, to know the alphabets, not only to know the alphabets, but the sequence of alphabets, the arrangement of these alphabets, A, T, G, C in our chromosome. So this was a very ambitious project, which was started in 1985, 1986, and subsequent governments, they invested in this project because they considered very important. And the first paper on first scientific paper was published on Human Genome Project was in 2003. It was a big journey, big investment, but what is unique for this project, what happened that NIH and a private company, Biotech, they came together, Celera, they came together to discover the Human Genome Project. So Human Genome Project, one of the most ambitious scientific project that was done with the government as well as with Celera Genomics, right? And why it is important, for example, a lot of people who have disease in their family, they want to know early, because the earlier we know, we can institute treatment. So how to go? We don't have the symptoms, but we know there's a family lineage, so it's better to know. So that's the advantage of Human Genome Projects. And science is interesting. There's no substitute for science and scientific development, but yes, we must look at it from both the sides, the pros, the positive and the negatives, so that we are aware and we are ready when it comes to decision-making time. So with this, I finish this small chapter also, which is made up of several small topics, namely diagnosis, gene therapy, vaccines, regenerative medicine, Human Genome Project, and ribozymes. Okay, so take care. Thank you.